All right, we're here to look at linked genes and the work of Thomas Hunt Morgan in this slideshow. And when we're talking about genes, what we're talking about are the specific sequences of the DNA that are lying in the chromosome. Hopefully remember from a couple chapters ago that chromosomes are just really densely coiled bits of chromatin, and chromatin is just the long strands of DNA. And a locus is a location on the chromosome. So it's basically, if you look at the chromosome as kind of like a pool noodle, it'd just be like one chunk of that pool noodle, transfer section of that pool noodle, that would be kind of like a locus. And what Thomas Hunt Morgan figured out was that, you know what, Mendel's not always right. Sometimes independent assortment isn't happening, and sometimes these genes are linked. So let's figure out how Thomas Hunt Morgan did this. What he did was a test cross, and hopefully remember that a test cross is when you take an unknown organism that shows a dominant phenotype and made it with an organism that's showing recessive phenotypes, because we know the genotypes of the organism showing the recessive phenotype, because in order to show that recessive phenotype you have to be homozygous recessive. So he actually did a dihybrid cross to figure this out. He looked at a fly that had a wild type body and a black that was heterozygous for a wild type body and heterozygous for wild type wings, and the recessives of each of those is having a black body and a vestigial wing. And when he did this one that he probably didn't know was heterozygous at the time until he did the test cross, well, he should have expected to see a 1-1-1-to-1 ratio. He should have seen even phenotypes for all of them. So there's our wild, there's our black body vestigial. So these are the two that he crossed, and he made about 2,300 offspring. And based on Punnett squares and laws of independent assortment that Mendel brought us, we should have seen this 1-to-1-to-1-to-1 ratio in the offspring. Well, here are the numbers Thomas Hunt Morgan observed, and most of them showed the parental phenotype, which was gray and normal and black and vestigial. So most of the flies came up looking like this. But there were also some recombinant phenotypes, where you had the gray mixed with the vestigial wings and the black body mixed with the normal wings. Realistically, we should have seen even numbers for all of these, but we saw more of the parental phenotypes being shown together and fewer of those recombinant phenotypes. So, in conclusion, Thomas Hunt Morgan started to realize, you know what, this is challenging this idea of assortment. So he came up with the idea that independent assortment isn't always accurate, because sometimes certain genes and certain traits are linked together. And when we talk about phenotypes recombining and genes recombining, hopefully remember that happens during crossing over in prophase 1 and meiosis. If you remember what goes on in crossing over, basically chunks of homologous chromosomes swap spots. So you have one set of chromosomes from your mom, one set of chromosomes from your dad, and the homologous ones would be the ones that code for the same information. During crossing over, a chunk of your mom's chromosome would switch over with that of your dad's chromosome, and that would sort of mix up the alleles. And sort of here's what we sort of got going on here. He's seeing a mother's chromosome and a father's chromosome. They form that tetrad during prophase 1, and the legs sort of mix up, and that can create some recombined genotypes, which would lead to a recombined phenotype. Geneticists use these recombination frequencies. So they look at the offspring and figure out what's the frequency of offspring that are recombined. The more often something recombines, the more often it gets mixed up, that tells us that those genes are farther away on the chromosome. The less often they get mixed up, that means the closer together they have to be on the chromosome. And that leads us to this idea that more common recombination means the loci are farther apart. Less recombination means the loci are closer together. So if we look at this example of a chromosome over here on the right, yellow body and white eye are by far the closest traits, that genes that are on the chromosome. So they're going to recombine the less. So more often than not, you're going to see yellow-bodied flies with white eyes, rather than yellow-bodied flies with vermilion eyes, because yellow body and vermilion is a lot farther apart on the chromosome. So there's a better chance that yellow and vermilion would get mixed up if that were the case. This is all a result of crossing over, and by using these recombinant frequencies, geneticists can actually map the genomes of organisms, and this is what Thomas Hunt Morgan did that was absolutely genius. he figured out these frequencies and he mapped out 2,000 genes on the fruit fly's chromosome, which is pretty wild. In addition to mapping out and figure out genes were linked based on autosomal inheritance, he figured out some other stuff. Not only do organisms have autosomes, which are the chromosomes that code for pretty much everything going on, they also have one set of chromosomes that specifically tells you whether they're going to be a male or female. Females tend to have two chromosomes that look the same, so he named them XX. Males have one X chromosome that looks similar to the female one, but they also have another one that's a little different shape, and that one is called the Y chromosome. Usually, there's not very much information on the Y chromosome aside from how does an organism make sperm to reproduce. So usually when we talk about, all the time actually, when we talk about the Y chromosome and sex-linked traits, we say that the Y chromosome is sort of a blank. It doesn't really hold any information at all. And what Morgan figured out is that there's certain information carried on those X chromosomes for the flies. And when you look at the sex chromosomes, it's only the X carrying the information. So here's the cross he noticed. He said, if I took a red-eyed female, notice we got the two X chromosomes, with a white-eyed male, X and a Y chromosome, and I cross them, well, this is the offspring I get. I get all red-eyed flies. Now, even though the male had white eyes, none of the other males have white eyes because the male fly that provided the sperm to the female only can give other males the Y chromosome because that's what makes male flies males, having that Y chromosome. The X chromosome that had the white eye trait got passed on to the daughter flies, and they all became carriers of that white eye trait. Now, every so often, it's not nearly as common, but he'd find a white-eyed female. When he found a white-eyed female and crossed them with a red-eyed male, you see the red-eyed male up on top, white-eyed female over here on the side. Notice the progeny, or the children. All the females had red eyes. Only the males had white eyes, and every male that was produced from a white-eyed female and a red-eyed male had white eyes. So this led him to believe that, you know what? There's these certain genes that are linked to traits, and he figured out eye color in flies was a big one. If we're talking about humans, red-green color blindness is something that's caused by a sex-linked trait. Now, when you look at this pedigree, we see 1, 2, 3, 4 generations. Do you see any females that are affected as being colorblind? Well, why don't you think the females are affected? It's probably because in order for a female to be affected as colorblind, they have to have a male that's colorblind and a mother that's either colorblind or a carrier for the colorblindness trait getting together because of the XY relationships when you do the Punnett square. Females are more often than not going to be carriers for these recessive X-linked disorders because females have to be homozygous recessive to show it. And the only way they can do it is if a male is colorblind and the mom either has the trait or is colorblind and gets together. Now, there's a lot of female carriers in here. Notice all the males are either have colorblindness, like this one and this one and this one and this one, or they don't. None of the males are carriers because males are what we call hemizygous. Since the Y chromosome doesn't carry any information, you just need to have one colorblindness trait on that X chromosome in order for it to show in the males, which is why colorblindness is much more common in males. Another example of something that is an X-linked trait or sex-linked. X-linked and sex-linked are the same thing. Any other kind of inheritance called autosomal inheritance, just so you know. But hemophilia is another trait that is X-linked, which is another thing where when we look down at this pedigree of the royal families, we got some British stuff going on over here. We got the Germans, we got the Russians. Everyone in Europe was getting involved in this family. But we got all this royal pedigree going on here. The females are the carriers, and the males actually show the hemophilia, which is a blood clotting disorder, which basically if you fall down and get scraped, you won't stop bleeding unless you take some drastic measures to make it stop. The last type of genetics and gene passing that we have as humans is our mitochondrial inheritance. Your mitochondria has DNA. There's also things called plastids. Your chloroplasts are plastids. They also carry their own DNA. And we sort of hit on this a little bit earlier in the semester, but scientists think that mitochondria and chloroplasts were their own organisms at one point in time. And they found some eukaryotic cells, and they decided to live with those eukaryotic cells. And the mitochondria provided energy catalyzing reactions inside the eukaryotic cell, and the chloroplast provided reactions to collect sunlight and create food for the plant cells. Well, we think they were their own organisms, and through this idea of endosymbiotic theory, they started living with other eukaryotic cells, and something that's been passed on to us over the course of many millennia. Now, the mitochondria for you and I are only passed on from your mother, so we call that, this is also known as cytoplasmic or maternal inheritance, because mitochondria live in the cytoplasm and they're passed on by your mother. The only thing the father really provides to you and me is sperm with DNA. You basically get the nucleus from the father that combines with the nucleus in the mother's egg, and then pretty much everything else comes from the mom. So mitochondrial DNA is passed on through what we call maternal inheritance, and that should get us through the genetics section of this chapter. Hopefully you're enjoying.